dear directors, dear professors, dear students, and dear friends in this audience. I'm particularly happy to be here today and to discover this fantastic institute in Pune and discover for the second time India. And I thank, of course, all the responsible person, including CEFIPRA, for the invitation, and also our colleague now for this warm introduction, in which, in fact, he has said most of what I'm going to say already. So you will be at ease, I guess, if my presentation is not accurate. So let me first tell you what I want to do. Actually, you know now that we work in Drosophila, you know that we work on some types of receptors. I would like for the younger people in the audience to show them how we got to the state where we are now, what we think we understand in the system, and also to encourage them to continue in this direction. This morning I had the pleasure to be with a large group of students, and they asked a lot of questions. And nearly half of the time I was going to say, please wait until this afternoon, because I'm going to answer the question in my presentation, because I had foreseen to give a certain number of answers to very frequently asked questions. Question number one, which I got so often over the last year: why do you work on insects? I mean, everyone is interested in human health, aren't you? Of course I am. So why then not work on humans directly, and why work on insects? I will first explain in 2 slides how this came about. So let me show you an insect here. Insects are frequently infected by both fungi, bacteria, viruses, protozoa, just to Plasmodium. And when I grew up in Luxembourg, Echternach, as my colleague said a few moments ago, I was the son of a zoologist, professor of zoology, as shown here, my father, Jules Hoffmann, and he spent all his spare time, he was a teacher, spent all his spare time studying insects. He was very interested in the physiology of insects and the systematics of insects, and he got me interested in zoology and in this group. And as I said this morning, answering a question, without my father, I would probably have been in literature and humanities. So it was a forced motivation. And he was a very strong personality. And then I moved to Strasbourg, and in Strasbourg to do my university studies there. Luxembourg had no university at that time. And I met a professor whose picture is next, Professor Pierre Joly, who worked on grasshoppers. And I started my work on grasshoppers, and the rationale was the following. At that time, France was strongly involved in what I'll call responsibilities, whatever that is, in Northern Africa. And there were big problems with grasshoppers there. And it was suspected that the passage from a gregarious phase to a solitary phase, or vice versa, was controlled by hormones. And Professor Joly and other people suspected that these hormones were really the clue to the passage to the swarm-forming groups of insects. And so what they had done was continuously transplanting endocrine organs from one insect to the next. And they had done this over 25 years. And as he reflected to me when I asked him for a PhD subject, he had never seen the appearance of any opportunistic infection, although they didn't take any precautions at that time. So he guessed that the insects would have very strong antimicrobial defenses. So, insects are a very important group, as you may know in this country. Also, 80% of all living species in the world today belong to the group of the insects. They destroy 1/3 of all human crops annually, and they put 1/3 of humanity at risk of diseases for transmitting parasites, viruses like dengue, and bacteria. So this appeared as a very challenging topic, a very large group, important for mankind in a certain sense, and very strong, potentially very strong antimicrobial defenses. So I was very excited to start this topic. We didn't know what to expect. Frankly, we didn't know. I certainly never thought of medicine at that time. I had even decided that I would not study medicine. And so I'll take you shortly through, in the next 40 minutes, take you shortly through the way things evolved. So what we knew, it has been mentioned already by my colleague a few minutes ago, we knew from the work of Metchnikov, that invertebrates, insects in particular, and also vertebrates, respond to an infection by a cellular defense mechanism which is called phagocytosis, which corresponds in the engulfment of bacteria by a dedicated blood cell type, which then destroy the bacteria. And we also knew from work which had been forgotten in the literature. Metchnikoff has never been forgotten, but the other people here, Payot and Glaser, have been totally forgotten in the literature. And we were able to dig them out in our library in Strasbourg, their work. They had shown that there's also an aspect of production of antimicrobial activity appearing in the blood. But it stopped there because at that time, of course, the techniques were not available to go further and identify these molecules. So when I started on the grasshoppers, the first thing we did was to challenge those with a low dose of bacteria, and then to see if we increased their resistance to a second dose, and indeed we did, even to a lethal dose. But then, when we went into the detail of these experiments, we saw that this was simply by stimulating the production of blood cells, hematopoiesis. We couldn't find at the time any evidence in the grasshoppers, any evidence for the induction of any antimicrobial, soluble antimicrobial activity in the blood of the grasshoppers. Sometime later, we turned, so I did my PhD work on the hemopoietic tissue of the grasshoppers, and from there on went into two different directions, which became complementary later on, that's to say development and antimicrobial defenses. But let me go shortly, because I'm not going to speak until dinner time, let me go shortly now to what we found about the inducible antimicrobial activities. So in this case, I'm illustrating here a Drosophila. But initially, in the 80s, we couldn't work on Drosophila because our physical-chemical methods of identification of potential inducible antimicrobial peptides were not accurate enough, were not refined enough to work on a small insect like Drosophila. So we worked on large blowflies, and we looked at the activities present in those blowflies, and we extracted first those molecules. And I'm illustrating here what you can see in the fly, in Drosophila. You could also see it in blowfly. You cannot see it in grasshoppers. We can discuss this later if you have time. So what you see is that when at time 0 you prick a fly with a needle which you have dipped in a bacterial culture or fungal culture for that time, or mix, you see that with a very simple growth inhibition assay, you note the appearance of antimicrobial activity. And that has also had been noted in butterflies by Hans Boman, who was in Stockholm at that time. And he was on the PhD defense committee of my wife, who worked with me at that time, and I'll show her a picture a little bit later. And he invited her to come to Stockholm and use this assay, working again, still on butterflies, Galleria at that time. Now, we in the group asked 3 questions. Number 1, what is the identity of the molecules which are induced? Following the work of Hans Boman on butterflies, Indo-French Cecropia, we suspected that in the fly we would find antimicrobial peptides, that the nature would be peptidic. I'm sorry. Second question, in the case that these are peptides, how are the genes encoding these peptides controlled? How is the expression controlled? And the third question, how does the insect notice that there is an infection, and can it discriminate between various types of infections? So those will be the 3 questions which we'll ask in the fly, and to which I'll come back in the following slides, and I'll try to compare with what we have learned in mammals. Now, to make a very long story short, it took us several years and many efforts to find that in the fly there are at least 7, now we could go up to 8, families of inducible antimicrobial peptides. They're shown in this slide, and they are essentially, not exclusively, but essentially produced in the fat body of the fly, which is some sort of equivalent of our liver. I've noted here dipterosin, which was the first molecule which we identified in large blowfly. It's anti-gram negative. Also noted that we have, through homology cloning, we have found etacin and cecropin, which are similar to the molecules found in the butterflies by Hans Boman. We discovered a new molecule which we call trozosin. All those molecules are active against bacteria, gram-negative bacteria. Then we found metchnikoffin, and which is defensin. I've shown you defensin, I'm not going to comment listericin, which has been recently found by another group, but we found defensin, which are active against gram-positive bacteria. And finally, and this was really a watershed in our laboratory, I'll present it here as I have this slide. We were at a given moment, we were at the end with our methodologies of the list of peptides which we could identify with the growth inhibition assay. And then we turned to comparative analysis of chromatograms of challenged and non-challenged flies without doing a bioassay. And there, finally, we found a molecule which is shown here on the right in the upper part of the slide, which we found out, again, after 2 years of intense work, to be antifungal. We call this molecule drosomycin. I'll come back to drosomycin and to listericin in the next slides. Now, an important point was at that time that when we cloned the genes, and I should say in parallel when our friends in Stockholm in the Bowman laboratory cloned the cycropin gene, we found in the promoter sequences of these genes, we found binding sites to NF-kappa B. NF-kappa B is a central transactivator of immune responses in mammals and of stress responses in mammals, as known at that time through the work of David Baltimore. I'll show him in a second. And so a few years earlier, David Baltimore had found this transactivator and in kappa B cells and B cells of lymphoma. And so the question arose, would those sites be involved in the induction of the antimicrobial peptide gene? That is to say, in the transcription, the control of transcription of these genes. Now, the beauty of the fly allowed us, which we had introduced at that time in the laboratory in the late 80s, the beauty of the fly allows you to establish transgenic flies in which you can replace the coding sequence here by a reporter gene. This is like Z, which will give you a blue color in the presence of a colorless substrate. So what we did is we established these flies, transgenic flies, and as shown here, in an unchallenged fly, there's no reaction of this leg Z, this blue color, whereas in a challenged fly, it appears very rapidly, and you see everywhere in the blood, in the fat body cells, which are dispersed through the body in this adult fly. Now, again, as I said, the beauty is that you can now mutate it. You can establish transgenic flies which have mutated sites. You can mutate them the way you wish. You can even put more of these sites here, and you look then at the what happens, and that clearly showed us that if we mutated the sites, we could not induce anymore the expression of the antimicrobial peptides. So in other words, these peptides, these motifs, these response elements in the promoter sequences of all the antimicrobial peptides as we know now, and of all the immune response genes as we know now in the fly, are dependent on the presence of wild type NF-kappa B binding sites. Now, excuse me. So it so happened that we now know, and I'm just summarizing here information which was gathered over the years. The fly has 3 members of this NF-kappa B family. We have 5, and they are shown here. There are 2 types, like in humans. This type here, the names are not relevant for us at this time of the presentation. They have all the relevant homology domain, which is the domain which is going to bind to DNA, and they are retained in the cytoplasm by binding to the inhibitor. Again, let's just keep the term inhibitor. And those 2 molecules are kept by the inhibitor. And a longer molecule, which was named Relish by Dan Haltmark in 1996, has these inhibitor domains in the C-terminal part of the molecule. This needs cleavage by a dedicated protease. So let me now just explain in very simple terms the role of NF-kappa B in any system, as we know now, mammalian as well as in the fly system. This is David Baltimore, and I've put here, this would be a mammalian NF-kappa B family member, and this is the inhibitor. Now, that's in 3D structures, which is work from several laboratories over in the early 90s. We see this is NF-kappa B shown here, and it is bound to I-kappa B, the inhibitor. So the only thing what you need now is remove I-kappa B, and this is normally done by phosphorylation of NF-kappa B, and then, the inhibitor I-kappa B, and then NF-kappa B dimerizes, goes into the nucleus through a nuclear localization signal, binds to DNA, and controls expression of the genes. The beauty of this system is that you have everything in place at the time of the microbial aggression. Everything is there, and you can, in a matter of seconds, you can, by just removing the inhibitor, you can have the nuclear translocation of NF-kappa B and the induction of gene expression. And now there was, at that time, when we became convinced that NF-kappa B was involved, there was a study which had been published several years ago by a group in Germany, headed by Christiane Nusslein-Volhard, was awarded the Nobel Prize in 1995, together with Erich Wieschaus and Ed Lewis. And what they had done in that laboratory was to feed a mutagene to adult flies and to look at the progeny and to screen for abnormal embryos. And those abnormal embryos, they gathered, would have an effect, a deleterious effect in genes which are responsible for the organization of the dorso-ventral axis of the fly or the anterior-posterior axis of the fly, or both. Now, the work of Nusslein-Volhard, which I'm going to summarize shortly here, has pointed out that there was one transactivator present in the cytoplasm bound by an inhibitor. Again, I'm not mentioning the names, which refer to phenotypes of the abnormal embryos. It turned out, through the work of Stewart in '86, that this molecule, dorsal, is a member of the NF-kappa B family. It turned out that cactus, shown here, is very similar to mammalian inhibitor I-kappa B. Now, what they found in the large screen, which took years, they found that this system became activated when a transmembrane receptor, which they dubbed TOL, in reference to the very peculiar phenotype of the dying embryo, which resulted from their mutagenesis screen. So I'm not going into these adaptoproteins and kinases. So she pointed out that TOL was required, the TOL gene product was required for the induction of this system, which then led to the transcription of embryonic genes involved in the embryonic development. Now, it turned out in this screen that TOL was activated via a cleaved form of polypeptide, which is in, we're speaking here of perovitamin space, and there's a very complex cascade of proteases here which become activated in a way which is still not fully understood in this system. So, this begged, of course, the question. We knew we had a very strong inducible system. We knew that it certainly was mediated via NF-kappa B, or the likelihood was great that it was mediated via NF-kappa B. So, would this cascade of Nusslein-Volhard and her associates, would this be involved in the immune response? The odds were against the hypothesis, for the simple reason that all the genes which were identified in this study were considered maternally expressed genes. And it was objected to us that if this were the case, then of course the embryo would produce all the antimicrobial peptides and so on and so on. But when we looked at this, it appeared clearly that the genes were expressed indeed later in development in the fly, even in the adults, and that challenge, an immune challenge, would I'm not finding that word now, would increase dramatically their level of expression. So, we decided we would look at this, and this took quite some time. It took several years. We had at that time, we had Bruno Metterjohn, who was just had defended his PhD thesis, and he was trained as a Drosophila geneticist. And so what we did, what he did in this case, was looking at the induction of the expression, let me show this here, of the expression of the tyrosine gene, which at that time was the only one which we had in our hands. And this is looking at, this is a simple normal plot, this is looking at non-infected, non-infected flies, and this 6 hours after infection, and you see that the tyrosine gene is very nicely increased, as we had anticipated. These are loading controls. Now, the great disappointment came when we looked at TOL deficient flies. At that time, we were looking at mutants for the dorsal NF-kappa B family member, which we had in the laboratory, and in this case, we saw that the system still remained fully inducible. This was a black moment in the laboratory, and that's why I mentioned it for the young people. So you think that you're progressing, that everything becomes clear. You do the key experiment, and it turns out that no. you were wrong. Something is wrong here. And the way we came out, we overcome this was not, this time was not through genetics of the fly, but it was through the work which I've mentioned of another group in the laboratory who were biochemically oriented and had done an analysis of all the peptides or polypeptides which were induced after challenge. And it took them nearly 100,000 flies to do this work. And I will always remember when it was done, because they dubbed this experiment Drosophila, in reference to Schwarzkopf, a desert storm operation at the time in Iraq. So Drosophila then yielded a large number of inducible polypeptides, among which one was particularly prominent. And this polypeptide, as I already mentioned, had no antibacterial function. We suspected that it would be probably protease inhibitor, given that we suspected proteases to be somehow involved in the activating cascade of toll, although we couldn't show that toll was there at that time. So we were trying everything we could. And it was thanks to a meeting where I met a professor from Leuven University in Belgium who worked on plants. And this man had found an antifungal peptide in plants. And when I saw his sequence on the slide on the screen, this was at the Stockholm meeting of the European Society of Biochemistry. It looked very similar to the structure which we had established already for our molecule. And then we teamed together and we could show that the Drosophila novel molecule was indeed antifungal. Then we named this molecule drosomycin, and drosomycin changed the view in the laboratory, because when we went back to look if drosomycin was inducible, in this context, you see the result was clearly, it was inducible, of course, by challenge, but it was non-inducible in the toll deficient background. Excuse me. What is more, when we looked at cactus deficient flies, remember cactus, which I did not use in earlier slides, cactus is the inhibitory molecule. So when you take away, which you can do by genetics, there are mutants, loss of function mutants in the cactus gene. When you take that molecule away, you would anticipate that now NF-kappa is not retained anymore in the cytoplasm, goes into the nucleus and controls the transcription of the gene. And this is exactly what happened. Drosomycin, in the absence of a cactus, without any infection, is constitutively expressed. So this was then, after the dark moment 2 years earlier, was a great moment which came. And also, interestingly, we saw that dipterosin was not induced in this context. So there was a big difference between the inducibility of dipterosin and drosomycin. And fortunately at that time, for reasons which will be too long to explain, we had a mutant fly which was deficient in phenol oxidase, which is one of the arms of the insect immune defense, and which we named IMD for immune deficiency. And in this context, we had the converse phenotype, that is to say, drosomycin remained inducible, but dipterosin was not. And this led us then to propose that the fly had two distinct pathways controlling the expression of antimicropeptides, or basically doing this. One was the partial reuse of the toll pathway. Not all the genes of the embryonic system are induced in this system. Another was what we proposed to call the IMD pathway, with Bruno Lemaitre and Jean-Marc Reichard and two students, and which at that time, which was a little bit courageous, because at that time we only had a mutant fly in our hands. We hadn't cloned the gene, and we hadn't done any rescue experiments and so on. But, so rapidly now, let me say that during that period in '91, which has already been mentioned, I had contacted Charlie Janeway. At that time, he was at Yale University. He has passed away a few years ago. And he was very much interested in innate immunity. Let me say, to be more precise, he was interested in the activation of adaptive immunity, which he suspected would come from innate immunity. We also had teamed up with Alan Zykowitz at Harvard Medical School, and who was still a very close friend of ours, and who was interested in the mannose-binding lectins and the mannose-binding activation of complement cascade. And some of you may be aware that activation of complement can require a protease cascade like the one which is which I've shown for toll in the embryo. So what they asked when they saw our results was, how does this translate into everyday life for the flies? They say, are those pathways really required for the survival? And as shown in the following slide, this is infecting a fly, wild-type fly, red line with E. coli, gram-negative bacteria, and IMD mutants with gram-negative bacteria. IMD mutants, remember, produce the antibacterial peptide dericin. The IMD pathway controls the expression of the antibacterial peptide dericin and other antibacterial peptides, and you see that there's a very big difference in the survival rate. Really, the IMD pathway does play a role in the survival of the fly. And in the converse experiment, we were looking at tall mutants infected here by Aspergillus, and as you see, the Aspergillus will kill the flies after a given time interval, but the mutants are already, half of mutants are dead after 2 days. So the survival is very severely compromised. And this then gave this slide, which has made its way unknowingly into many textbooks in which we looked at, this is a picture of a fly which has been challenged with Aspergillus. It has died and it is overgrown with hyphae, and the reason we wanted to show this is that it really is a demonstration that the toll pathway prevents any resistance to those fungi. Now, this was in the history of our laboratory. This was an important moment. And let me shortly explain to those of you who are not hardcore immunologists why this had relevance far beyond the fly. So I'll take 2 slides to show the mammalian system and to give an introduction to the mammalian immune response. The first slide shows a phagocyte, and phagocytes are not restricted to mammals or to vertebrates, but they're also present in insects. They're present in every group as we know now. And they are the basis of innate immunity. They would be the cells of the time of Medvedev. Now, innate immunity, which was not on the foreburner in immunological research in the up to, I would say, 10 years ago, is an immediate reaction. It has no memory, and it is common, as we now know, to all organisms. In contrast, adaptive immunity, which is shown here, there's one lineage here which is responsible for adaptive immunity, and those are the lymphocytes. They come under 2 forms, the B lymphocytes, which we know to produce antibodies, and the T lymphocytes, which are responsible essentially for cytotoxic T lymphocytes, shown here killing another cell. Now, the adaptive immunity is a slow reaction. It takes several days. Now, the problem which we all face is when we have an injury. Let's say, if I address the male part in the audience, if we cut ourselves while shaving in the morning, we're surrounded by fungal spores, surrounded by bacteria, and there will be an opportunistic infection going on nearly immediately. So we need a defense, an immediate defense, and we cannot wait for several days. So adaptive immunity is a slow reaction. It is found only in vertebrates. I'll come back to this a bit later. It appeared with the lymphocytes, which go together. Lymphocytes appeared only in the vertebrates. They are absent from all the invertebrates. There's a large repertoire of receptors, and this was a very interesting topic, and this was a topic which was really the center of most of the interest in all of immunology over the last 50 years. And several Nobel prizes were given to studies on the production of this large repertoire of receptors. Now, the interest of this system is that it gives memory cells, and consequently, it allows the possibility for vaccination. Now, the next slide shows us the question which was central in the mid-90s in the discussions, and that was the tenet by many people, but it was very nicely written down by Charlie Janeway in 1809, that adaptive immunity needs an input from innate immunity. So here I've shown the dendritic cells. So, dendritic cells are the paramount cells of the innate immune system in mammals. They have been studied in great detail by Ralph Steinman, who was associated with Bruce Beutler and me last year. Now, in this system, What was known is that activation of naive lymphocytes by dendritic cells of the immune system requires the role of what was called stimulatory co-stimulatory molecules. MHC presenting the peptides, this will be the major histocompatibility complex, and here the T-cell receptor. I'm not going into detail, but let us just keep in mind for the whole audience that NF-kappa B, this transactivator, which we now know to play a role in the fly, which was known to play a role in mammals, NF-kappa B controls the expression, or increases the expression of those molecules required for activation of naive lymphocytes. Now, what activates NF-kappa B? It was supposed that it was a receptor, an unknown receptor, and microbial fragments would interact with this microbial receptor and then stimulate the system. Here we have phagocytes, which are taking up dendritic cells and which downgrade here the peptides in this bacteria and then present them for the MHC to naive lymphocytes and so on. And the result of this activation is doublefold. Number one is immune response genes, namely the antimicrobial peptides, are induced in response to the stimulus from the dendritic cell or macrophages or monocytes. And dendritic cell is not the only antigen-presenting cell, as we all know. Now, this type of response is the one which we found in the fly. And this type of response was later on also found in mammals, and it is now, it's now a center of very much interest in mammals. We know that we produce large amounts of antimicrobial peptides. For instance, the figure goes that on our skin, we produce 10 grams of antimicrobial peptides and per day. So showering is not always recommended, not too often. And so in addition, very recently, a group in Holland has found that we humans produce a drosomycin-like protein. See how it is a small word, as people say. Now, the second effect downstream of NF-kappa would be the activation of adaptive immunity by innate immunity via the production and the stimulation of those so-called co-stimulatory molecules of the expression and the production of cytokines. So there are two essential effects in mammals. This was sort of perceived to some extent in the mid-90s, but the problem was that no one had identified the receptor, and the receptor many groups were working on this receptor, and including then, of course, at Charlie Janeway and then at the Scripps Institute in many laboratories. And essentially, people were looking at the possibility that lectins could be involved. And this was also our first idea in our collaboration then with Alan Zikovits and Charlie Janeway. Remember that Alan Zikovits worked on mannose binding lectins, which activate complement. And so at a given moment, we were interested. Everyone was sort of excited about this idea. But when we look now at the toll receptor, and the structure of the toll receptor, which came out, as I mentioned, in the late 80s, it appears to have an extracytoplasmic domain, which is very similar to the one which was found at the Scripps Institute by Richard Yulovich in the receptor CD14 is a receptor for bacterial lipopolysaccharide. However, this molecule is GPI anchored, so it cannot signal to NF-kappa B. And as you remember from the preceding slide, everyone was looking for a receptor which would be able, upon stimulation, to signal to NF-kappa B. Now, on the other hand, bona fide stimulator of NF-kappa B is the interleukin-1 receptor, which is shown here, which has a distinct domain here. It has immunoglobulin-like domains outside in the extracytoplasmic portion, but it has an intracytoplasmic domain which, just to believe the color, is very close to that of a toll receptor. So this boils down then to say a very simple take-home message would have been, and was in 96, that at least in one instance, nature has chosen a chimera of an extracytoplasmic domain, which can play a role in binding to lipopolysaccharide, and an intracytoplasmic domain, which in a different system can activate NF-kappa B. So two domains, which in distinct systems had been functionally analyzed, appeared here to be present as a chimera, and above all, this system in the fly activates NF-kappa B and is responsible for the antifungal defense. So, of course, the first time I showed these results, was at a meeting which we had with Charlie Genoway and Alan Zickovitz near Boston. We were at that time supported by a common grant from the Human Frontier Science Program, and have ever since been supported very generously by the NIH. And so the year after Charlie Genoway and his new associate, Rasul Medjitov, went to look for a human homologue, and indeed they found a human homologue, as illustrated here. This has already been mentioned by the previous speaker. In 1997, they found that there was a human homologue, and it signaled activation to adaptive immune response. Was this the end of the story? Not truly, and this has already been mentioned, because it was difficult for the group of Genoway to show the effect of a microbial inducer on the system. So they had transfected cells with an intracytoplasmic domain, which could activate the expression of interleukin-1 via NF-kappa B, which was fair, and they could not, they hooked extracytoplasmic domains of the CD4 receptor, but they could not get the system stimulated by microbial ligand. And this is why the story came to a close only the year after, when Bruce Beutler started out from a totally different perspective. Bruce Beutler had been working on tumor necrosis factor for many years at Rockefeller and then in Texas, and he was interested in finding the receptor of lipopolysaccharide, which was known to strongly induce TNF, tumor necrosis factor, which is responsible, as many neurologists may know, for the endotoxic shock, which still kills every year tens of thousands of people in hospitals in the Western world, and probably also in over here in the Eastern world. So he was looking for this receptor, and it took him 5 years. He went about through positional cloning in a mouse which was known to lack resistance to gram-negative bacteria, and which was known also to be resistant to the endotoxic shock. So lipopolysaccharide didn't do anything to that mouse. So it was gathered, it was supposed that this mouse was lacking the lipopolysaccharide receptor. When Bruce finally, with his colleagues, when they finally cloned this receptor, again, I say after 5 years, they found that it was equivalent and instructor to the one which had been published the year before by Medjitov and Genoway, and was similar to the one which we published 2 years earlier in the Fly. And then the whole system became, there was a whole explosion in the system, which is illustrated by the fact that ever since there have been about 20,000 papers published on toll receptors in various situations, particularly, of course, in human health and in mouse. And I'm just going to summarize the information rapidly in the next slide. We now know that we humans have a dozen of receptors, of toll-like receptors. Mice have an equivalent number. Not all are functional either in mouse or in humans, but I don't have time to go into this. And we have toll-like receptors on the cytoplasmic membrane, as illustrated here, and they all interact with microbial cell wall components, like the famous lipopolysaccharide, like lipopeptides, flagellin, and so on. And inside the cell, but not in the cytoplasm, we have in the endosomal compartment toll-like receptors which bind to RNA, single-strand, double-stranded, or to CpG DNA, which is characteristic for bacteria. Now, as a result of this binding, the signaling domains, the intracytoplasmic signaling domains, here and here, will signal via various adapter proteins. I have no time to go into these, but there's many nice studies on these adapter proteins, which will sort of select what response is yielded in the end, and they activate NF-kappa B. They activate NF-kappa B, and the system activates NF-kappa B, and then goes to, as I already mentioned, to production of immune response genes, among which, but not only, antimicrobial peptides in all the systems which have been analyzed so far, and then, as mentioned, the activation of adaptive immunity. In mammals, and I should say in vertebrates, there's in addition another important transactivator, in addition to NF-kappa B, which is IRF for interferon regulatory factors, which have been worked out in quite some detail, and which essentially they play an important role in the immune response, but they are absent from the Fly. And I will not we have made no contribution to this aspect in our past. Now, let me then shortly say, in order not to make my presentation too long, I've taken away a series of slides and summarized the information which was obtained over the years about the fly system. So in mammals, I told you, at least in mice and humans, we have about a dozen of functional toll-like receptors. In the fly, the flies have 9 toll receptors. All fly toll receptors are involved in development. All, including the one which plays a role in antifungal responses. And why the fly has only selected one of its 9, we do not know. For immune response, we do not know. And incidentally, the first time we discussed this in more detail with Charlie Janeway and with Alan Zikovits, the idea came that maybe the fly, at that time it was known that mammals had different repertoires of recognition of microbial ligands between the various toll-like receptors. And the idea was that maybe toll-2 in the fly will recognize gram-negative bacteria. So far, I've only spoken about the fungi and gram-positive bacteria, and maybe and so on, but this turned out not to be the case. So far, to the best of our knowledge, only toll plays a role in the NF-kappa B dependent immune defense of the fly. Now, as a surprise, it came then that when after again, there were 2 years of work involved here by persons in the laboratory, it became apparent that toll was not directly binding, in contrast to the mammalian system, to microbial ligands. Instead, it was binding, as in the embryonic system, to a cleaved cytokine, which goes by the name of Spätzle, again referring to the phenotype of the mutant embryo isolated by the Drosophila group. So, toll in the fly is not directly activated, it's activated by a protein, by polypeptide, which is not, it's conceptually not abnormal because TLR5 in mammals is activated by flagellin, which is also a polypeptide. So, but how does this polypeptide in the fly system, how does it become cleaved? And there we could show, and others could also show, that there is a cascade, let me show, a cascade of serine proteases. So what happens, and again, sorry to repeat myself, but it took several years in our laboratory and in other laboratories also to decipher this cascade and to demonstrate that yes, toll is activated by the polypeptide, a cleaved polypeptide, and this is cleaved by a cascade of serine proteases, and they have all been identified now. They're not the same as the ones present in the embryonic system, so those are, it's another set of serine proteases, and the system becomes activated then when peptidoglycan interacts with a dedicated receptor in the blood. This receptor is called peptidoglycan recognition protein. It was initially identified in butterflies in Japan, and it is released by the fungus when it traverses the epidermis of the fly. Those proteases will activate, again, a dedicated serine protease in the blood, which we name Persephone, and this will then feed into this cascade. So you see, it's a relatively complex system. But at least I could say it is fairly well understood right now, with the exception of one point. We still don't understand how the binding of glucan to this receptor or the binding of peptidoglycan to this receptor will activate the cascades. So this remains one of the hot questions in the field. Now, let me now finally turn to gram-negative bacteria. And in this case, it is again peptidoglycan, which is an inducer. But this peptidoglycan interacts with, I'm not going to say dedicated, it will be the 5th time to say dedicated, but with a real receptor which is precisely, the role of which is to bind peptidoglycan from gram-negative bacteria. And here the beauty of the system is that, so this would be this receptor here is PGRP, so it is peptidoglycan recognition protein, so it's a family, there's a family in the fly of 13 such molecules. And there is a difference between peptidoglycan of gram-positive bacteria and gram-negative bacteria, and it's simply related to the 3rd amino acid in the stem peptide, which links to the sugar chains in the peptidoglycan. It's a DAP, diaminic acid in gram-negative bacteria versus a lysin gram-positive bacteria. And again, this has been worked out in structural detail, particularly by Hans Dijsselhofer in Dallas and by other people, Ian Wilson at the Scripps and so on. So we have fairly good understanding at the atomic level how these receptors can distinguish between gram-positive and gram-negative peptidoglycans. And so basically, this is it in the system. The recognition is, so the activation of the immune system in the fly is mediated by glucans, fungal glucans, and by two forms of peptidoglycan from bacteria. So it's a very simple repertoire, a very efficient repertoire, as you may imagine. So from there on, let me now very shortly turn to, so I think we sort of have understood now together what are the essential effector molecules. I've reduced my presentation to the antimicrobial peptides because they're easy to identify and the function is easy to analyze, but there are about 600 polypeptides which are newly transcribed, newly produced, I should say, in response to an infection in the fly, which is about half of the number which you would find in a macrophage, in a mouse macrophage. Now, so we have understood what are the effector molecules. We sort of know now how the genes encoding those effector molecules are transcribed. Now, let us turn, we sort of have, I was going to say, we sort of have understood how recognition occurs by the fly and via those receptors for peptidoglycan and beta-glucans. Now, what happens between the recognition and the transcription of the genes encoding the immune proteins? I'll be short on this. Not that it does not interest me. It's a very exciting topic. It's a topic which is moving quickly now, and I try to be personally involved in this. But as time is going way rapidly, is going on rapidly, let me just show the tool, what we know in a few words about the toll pathway, so which is activated by fungi, by gram-positive bacteria. I've made a point about microbial sensors, the protein cascade, Schwetzel is cleaved, toll is activated. Now downstream, there is an adapter protein, and please keep this name in mind. This is MID88, which is absolutely identical, absolutely close, I should say, homologous to mammalian MID88, which is an adapter protein. There's a kinase here, which goes by the name of Pella, KD stands for kinase domain, and an adapter, which goes by the name of TU. Now, here we have IFCPAR, and its inhibitor IFCPAR can be dorsal, which is not relevant to our discussion this afternoon. So what happens is that cactus will be phosphorylated and then degraded in the proteasome. Surprisingly, although many laboratories have focused on this, we still do not know the identity of the kinase. It is not Pella, which phosphorylates cactus, but we're working on that. And now, as a result, cactus, as I said, becomes phosphorylated and dissociated, degraded in the proteasome, and the NF-kappa family member with its rel homology domain, after dimerization, goes to the nucleus, controls the expression of drosomycin and hundreds of genes which concur to the defense against fungi or gram-positive bacteria. So we do not understand everything in this cascade, admittedly, and we're working on that, and I'm sure other people are also working on that. Now, let's turn to the MD pathway. The MD pathway has a special interest beyond that of the toll pathway, I believe, in a more general sense, because the MD pathway is closest to mammalian TNF receptor pathway. And you know, TNF, as I've already mentioned, plays a big role. It's the central, one of the central pro-inflammatory cytokines. It's not the only one, of course. And so we're very interested in the control of the MD pathway, which, as I mentioned, is initiated when gram-negative peptidoglycan interacts with this dedicated, I'll say the word this time, a PGRP here, and then we have a cascade. I'll be, again, short on that. We have IMD here. IMD is a DEF domain protein, and this DEF domain is closest to that of mammalian RIP. RIP stands for TNF receptor interacting factor. So you see we have here an adapter protein which is very close to one which we find in the mouse. So then we have FAP, which is identical to the one which we have in our own system. And this system will activate a MAP3 kinase, which is indicated here as TEC1. And I'm not going into more detail here, but TEC1 then will activate an equivalent of the mammalian signalosome, which has IKK beta and IKK gamma or NEMO homologs, which are here. So this is very similar with the absence of IKK alpha, which is present in mammals but absent here. Very similar to the one which you find in mammals. And now this is going to phosphorylate the big NF-kappa B member here, relish, which would be equivalent to mammalian P105 for those who are familiar with this system. And the molecule becomes cleaved by DRAD. And DRAD is a homolog of mammalian caspase 8. Now, in addition to what I've shown here, so cleaved Spätzle, cleaved NF-kappa B relish will allow the nuclear translocation of the rel homology to remain expression of the deutericin, and again, hundreds of genes in this case also, which will concur to fighting gram-negative. And the system is much more complex. And I have, of course, no time to go into this. Let me just point out, ubiquin is shown here. So the idea now is that maybe, so this is IKK gamma homolog is ubiquinated, as is this associating protein type 2, as is IMD. And the idea is that those ubiquitin chains will allow for the whole system to be brought to the cytoplasmic membrane when a stimulus occurs. And everything what we see now as activation may indeed happen upstream and then convey a signal to, even upstream we can probably have the cleavage of the NF-kappa B homolog relish in this system. Now, one important point, and this is sort of a message which I want to send to the various audience which I have privilege to speak to, the beauty of the fly system. We sort of say that this system, this IMD pathway is equivalent. I'm not saying homologous, but it's equivalent or evocative or close to or similar to the TNF receptor pathway. Now, the TNF receptor pathway, we are aware now, is activated also by endogenous ligands. Now we're passing out of the direct immune response to gram-positive, gram-negative bacteria and fungi. But in endogenous situation like misfolded proteins, like neurodegeneration, like arthrosis, everything which is related to, which can be related to inflammation, mutants, for instance, mouse or fly mutants of DNase 2 enzymes, which accumulate undue amounts of DNA, will end up by activating either the MD pathway in case of the fly, or the mouse TNF receptor pathway, as shown by Nagata in Kyoto a few years ago. So this system, and we find that in aging flies now, this system is activated in the absence of any infection. We find that during metamorphosis, when you have breaking down of various tissues, nearly all tissues, except for the imaginary discs, you have activation of this IMD pathway in the absence of infection. And even we find that in an Alzheimer's model of the fly, a Parkinson's model of the fly, which exists, in those models you have activation of the IMD pathway in the absence of any infection. So this really begs a big question. So what does activate in various situations this pathway? And to start doing this, we invested heavily saying much. We're a small group working with me around this question now. But what we have started doing was a proteomic analysis, looking at all the through EDIPRIC technology, which I learned this morning. We have obviously also here. We're trying to identify all the interactants. And the surprise was that it activated, this was in cells, in responsive cells, Drosophila cells, stimulated with heat-killed E. coli. We found that, let's call this molecule here the canonical members of the pathway, the ones which were identified through genetic analysis. We found with the proteomic analysis that we have 345 proteins associating within a few hours with these molecules. And of these, 90% are conserved between the fly and humans. And so far with RNA interference, which is not the best technique, which is a rapid technique, we have found that nearly half of these molecules are involved in the activation of the system here. So this is a lead system now, and we are working on this in association with Shizuo Akira in Osaka, and Bruce Beutler in Dallas, supported by the NIH. So we're trying here to say among those 345 members, there must be some. We know 90% are conserved between flies and humans. Some must be, well, they are mostly novel molecules of unknown, I should say, molecules, proteins of unknown function. And there must be probably a certain number of novel targets to play down the role of this activation pathway. Now, let me go a little bit further now, and in the next slide, what I would like to do, I realized I've forgotten to say that downstream of type 1, we have also activation of the gene kinase pathway. Now, in the next slide, I will try to compare what we learned over the years, what we learned in the fly, we and other colleagues in other laboratories who got interested in this over the years in the fly, and compare it to what has been learned during the same period in mammals. And as you will see here, this is a very oversimplified scheme, but it's enough for the point I want to make. So we have the toll receptor, the LG receptor. I'm not going into detail anymore, just let me point out again that we do not have yet identified a homologue of IKK complex. It may be that does not exist in the toll pathway, that it was lost during evolution. And let us compare now with the toll-like, the TLR4 pathway induced by LPS and with TNF receptor pathway. And you see we have here receptors which can in both systems be activated either by cleaved cytokine, in the case of Wetzler, or cleaved cytokine in the TNF receptor pathway in mammals, or direct binding to a microbial inducer like peptidoglycan in the pathway, or LPS in the TLR4 pathway. Then we have here the adapters, and you see MYD88 is present in both systems, in the fly here and in the mammalian TLR pathway. IMD is equivalent to RIP. We have the TEC1 kinase present in all the systems with activation of gene kinase, and we have this IKK complex. Here also it's a little bit more complex, if I may say so. In the main system there's still IKK alpha added to, which is absent from the fly here. And we activate NF-kappa B, and we go to the transcription of genes which are involved in immune response. Now, this slide is, for me, always brings an emotion. Because no one of us would have thought, no one in the world would have thought in the 60s or 70s even, that we would end up in the community of people interested in antimicrobial defenses, we would end up together and finding that the system is so parallel in flies and in mammals. Which of course begs the question, then, when did this system appear? And thanks to genome sequencing, which is now has become extremely wide over the world, and through genome mining, data mining, we now know that this system appeared very early in evolution. We have, I mean, all the members which I've painstakingly exposed to you in the fly are already present in the sea animal. They're present in crustaceans, in flies, cephalochordates, they have phioxus and so on, and they are present in the vertebrates, a point which I have heavily made in my presentation. So this system, this NF-kappa B based system, with toll receptors involved, with TEC1 involved, this system has appeared probably with multicellularity here about 1 billion years ago. And it is present in nearly all the systems. It has been lost occasionally, like in C. elegans. One type of worm has lost it, and this is C. elegans, a very peculiar organism. Also Hydra, a polyp, has lost it, but for the rest we find it in every detail. And even a question which I was asked yesterday in Delhi, what happens in the sea urchins? Sea urchins have 222 toll-like receptors. And so when that result was published, we said, well, it's obviously live as filters in the sea. That's not the answer, because Sayona, which also lives in the same conditions as sea urchins, has only 3 toll-like receptors. So you see, there's still a lot of things which you have to understand. Of course, when we speak about genome sequencing, we do not have the experimental demonstrations of the various functions there, and that will come over next years. And but for the time being, the only thing we can say that these molecules are present all over there. And in the sea anemone, there has been nice work done in Boston by a group Tom Gilmore and Bob Infinity, who have shown that the activation through microbes of the system IKB and FKB, IKB is really going on nearly like in the fly. So the important point to make here is that in mammals, I should say vertebrates, at one given moment, and this was probably around 450 million years ago, with the appearance of the placoderms, which are extinct cetaceans, that is to say cartilaginous fish, appeared the lymphocytes, and the lymphocytes allowed for all the processes which we know, the gene fragmentation, rearrangement of gene fragments, and for hypermutation, somatic hypermutation, which probably appeared a little bit earlier, but that's not the point. So my take-home message from this, before ending with 2 or 3 more slides, my take-home message from this is that we now understand that innate immunity is the response of 100% of all living organisms. I have been speaking so far essentially, uniquely about metazoans. There's similar things in plants, but we have no time to go into this. And so all metazoans share this innate immune response, and only 5% of the species existing today, and that is to say the vertebrates, in addition, have developed adaptive immune responses once they had differentiated lymphocytes from the myeloid precursors. And in this case, we vertebrates, we mammals, and we humans, we defend ourselves primarily by an innate immune response, and after 2 or 3 days, we will activate, via the innate immune response, we will activate the adaptive immune response, and this will give us this very efficient immune response, and also allow for memory, and saying this will allow for vaccination. So that was what I wanted to say about the work which we and others have done over the years, and which have led to really some sort of novel understanding of the way things happen in the antimicrobial defenses. A question which I often get is, how does this help in medicine? I'm not a medical person. I even think I mentioned decided with my father at the age of 18 that I would not take up medicine but become a zoologist. And you see I'm going through the back door into those various rooms. And, but still, just to make this clear for a more general audience, I have 2 or 3 more slides which address this question from the work, of course, of others with whom we have at least intellectually interacted. So TLRs, what are the therapeutic potentials? Very shortly, they play a role in vaccination as adjuvants, and this is one of the places where they can really become important. The understanding of TLRs in inflammation, in infectious diseases, in autoimmunity, in allergy, and in immunotherapy. Now, shortly, as regards, so this is, of course, a paradigm which I've shown already, so we're trying to understand the function here. But you will understand, to have a very good, very efficient adaptive immune response, you need a stimulation here, and to get the stimulation, you need an capability to be activated by these receptors, which are TLRs. So it is obvious, and then in addition, you have, of course, the antimicrobial peptide genes, which are expressed. So you understand how central these molecules are in our life, and how central they can become in medicine over the next years. So having said this, having said this, so what role do they play in adjuvancy? Now, in Europe, the essential adjuvant we are using is monophospholipid A, which acts via TLR4. In Japan, a lot of work is developed, as in California, around CPG oligonucleotides, which are DNA, parts of DNA, which act via TLR9. So what this tells us now is that many of the adjuvants are not very efficient adjuvants. You know that we still lack many vaccines. We lack good vaccines against, for instance, Plasmodium. We lack good vaccines against HIV, and even more generally, we lack good vaccines against many of the gram-positive bacteria, streptococci, staphylococci, and so on. And partly, people believe this can be due to the fact that our adjuvants are not efficient enough. So if you're a company now, if you know now which TLR receptor is involved, then you can play around. That's exactly what you're doing. You play around with the various types of adjuvants, and you try to find out the one which is most efficient without giving side effects. In the United States, it's aluminium hydroxide which is used, which is not. But the mode of action of that molecule is not known, but it's not acting, certainly not acting via TLRs. So the conclusion here is that some, but not all adjuvants act via TLRs, and this is presently a very important field of research and of innovation, and ongoing research for better adjuvants, and there is an ongoing research for better adjuvants, and TLRs represent the most promising avenue in this respect. This is the opinion of the people who I know in the field and who do work on this, which again is not what we do. So TLRs in autoimmunity, and I will, this is a problem which is, of course, very important also. Systemic lupus erythematosus, for instance, involves recognition via toll-like receptors of TLR7, namely of RNA, and TLR9 of DNA. And many of the autoimmune diseases have one component, at least, which is mediated by recognition through TLRs. In the case of multiple sclerosis, I'm told this is not proven. It's possible, but it's not proven. The same is true also for rheumatoid arthritis. Also, yesterday someone told me that he thought he had a demonstration that TLRs were involved in it, so, which I'm happy to believe. Now, TLRs and allergy, again, let me be short on this. The important point, so I'm not going into the detail of Th2 predominance, but the important point is that TLR agonists, let's say activators TLR, protect against autoimmune and allergic responses. And some of you may have heard about the hygiene hypothesis, which means that mediocre hygiene protects against overreactions of allergy. This is interestingly documented in Carelia, but maybe we can discuss that during the question session later. So let me then finally say that TLRs in inflammation, that is really the main role of TLRs. Inflammation is crucially important, dependent on TLR signaling, particularly in the context of infection. And this is the case in atherosclerosis, even in obesity, in Crohn's disease, and everything which ends with itis may involve TLR signaling. And the last point which I want to make, which is very important in my understanding, is that we have a certain number, we know now a certain number of genetic effects. And this is work, again, of my friend Bruce Butler, but also of Jean-Laurent Casanova at the Rockefeller Institute and at the Necker Institute in Paris, and in a certain number of small groups of young people coming specifically from some communities which are sort of inbred endogamy. There are sensitivities to some bacteria and or viruses, and they could be traced back to mutations in TLR4, in TLR3, in MYD88, in IRAK4, which is one of the single pathway downstream of TLR4, and ANG93B, for instance, just to give a few examples. So there are in nature, under normal conditions, we do have a certain number of patients which have a lowered resistance due to the mutations in these genes. Now there is admittedly a large redundancy, at least in our system, so mutation, lower expression of TLR4 will not automatically result in loss of protection because of the redundancy. But still in a certain number of cases, well-documented cases of children, this is the case. And with that, I would like to acknowledge the people who have been working with me over the years. I will start as a good husband with my wife, because Danielle Hoffman started with me. And so she stopped doing research a few years ago, and but she will come with me next year. Next time I come to India, next year, but she will certainly come. She loves the country. She was here independently with me one time. Jean-Marc Reichert is now the head of our laboratory, and he is an environmental biologist, now a Drosophila geneticist, and he has been part of most of the work which we have done over these many years. Bruno Lemaitre joined us in 91, I believe, and he has done excellent work in the Toll saga, Toll-Nandi saga. He's now working on gut immunity in Lausanne. Charles Hedrew has been with me so many years. He is a chemist by training, and he has been very influential in the work on identifying the various inducible antimicrobial peptides, together with Jean-Luc Dimarque and Philippe Goulet, also shown on the slide. And they were also at the origin of the company which we created to explore the therapeutic potentials of these inducible antimicrobial peptides. And Jean-Luc Dimarque is now in a high position in the company. Philippe Goulet is in Nobel. Dominique Ferrandon is an excellent geneticist, Drosophila geneticist, who did his PhD work with things like Pollard, I've shown on the slide. So he was ideally suited to come over to our laboratory. He has been here for nearly 20 years now. Julien Royer has worked on peptidoglycan replication proteins. He's now in Marseille. We are all group leaders or department leaders there. Jean-Luc Himmler is in the laboratory. He is going to be director next year of our structure. And he's the one developing the work on viruses. And I had, of course, no time to go into that. And we're what I believe, very interesting example of viral defenses on the time. And Elena Devashina from St. Petersburg initially, now a French citizen, working at La Charité in the Max Planck Institute in Berlin, is working on antiplasmodium reactions. She has been with us for 15 years, and we have a common international laboratory between her group and our structure in Strasbourg. I would like to end by also providing the names of people whose work I've integrated in those overall schemes from other countries, particularly the United States, Katrin Anderson, Karl Schmatter, and Neil Silverman and Tom Maniatis, Alan Zitowitz, not mentioned, and Nobel Perignon, and Michael Butros, respected Germany now, and David Schneider. In Europe, it's a group essentially in Stockholm, the group of Hans Bohrmann, I mentioned that he was from Austin. And then in Europe, there's other groups which are in connection with our laboratory, or which we were at the moment. And in Asia, I'm waiting to put more Indian names on this, but I'm sure this will come after my visit to India. This year we'll have collaborations here. And for the time being, this work is relatively well developed, essentially in Korea, and South Korea, and in Japan. And with that, I thank you for your patient attention. I'm sorry to have been so long. I was very happy to deliver this presentation.